afternoon, students. Today I am back with my next chapter, Cell Structure and Function. In this session, we are going to learn till prokaryotic cell. So, what do we know about cell? All organisms are composed of cells. Some are composed of a single cell, which are called unicellular, and others are composed of many cells, which are called multicellular. But whatever may be the case, cell is that unit of life which is capable of performing all the essential functions required to stay alive and stay independent. Thus, cell is the fundamental structural and functional unit of all living organisms, which means anything less than a complete structure of a cell. I'm repeating myself, which means anything less than a complete structure of a cell does not ensure independent living. Does not ensure independent living. So Antony von Leeuwenhoek first saw and described a live cell. Later on, Robert Brown discovered nucleus. The invention of microscope and its improvement actually led to the formation of electron microscope, which revealed all the structural details of the cell. Now let us come to cell theory. In the year 1838, Scladen, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which aggregate together to form tissues. Now, Swan, another British zoologist, studied different types of animal cells and reported that cells had a thin outer layer, which is actually known as plasma membrane. Nowadays, we know it as plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. And actually, we know that cell wall is very unique to the plant cells. It is absent in the animal cells. Swan proposed a hypothesis that bodies of animals and plants are composed of cells and products of cells. Now, Scladen and Swan together formulated the theory. This theory, although did not explain how new cells were formed, but still it is very critical to understand and know the cell theory. Then Rudolf Virchow first explained that cells divided and new cells are formed from pre-existing cells. He modified the hypothesis of Scladen and Swan to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and productive cells. All cells arise from pre-existing cells. Now let us understand an overview of a particular cell. See, cell is varies from different organism to organism and different types of organism. Why organism? It can vary even from tissue to tissue. Like in onion cell, which is a typical plant cell, it has a distinct cell wall or the outer boundary just within its cell membrane. But the cells of human cheek lacks this outer membrane and they directly have this cell membrane as the delimiting structure of the cell. Inside each cell, there is a dense membrane-bound structure called nucleus, which contains chromosome, which in turn contain the genetic material DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas if they lack a membrane-bound nuclei, they are called prokaryotic. I think we discussed about these things in the previous chapter. So you can go through my previous video where there is a clear, I think a table is given which shows a clear difference between eukaryotic and prokaryotic cells. Now, in both prokaryotic and eukaryotic cells, the semi-fluid cell matrix called cytoplasm is common. And this is the main arena of cellular activities in both plants and animal cells. And this actually occupies the volume of the cell. This occupies the volume of the cell. Cytoplasm is also the site of several chemical reactions which occur within the cell to keep it in the living state. Eukaryotic cells have other membrane-bound distinct structures called endoplasmic reticulum, Golgi body, lysosome, mitochondria, microbody, vacuoles, etc. But like nucleus, prokaryotic cell also lack such membrane-bound organelles. Ribosomes are non-membrane bound organelles found in all cells, which means both eukaryotic and prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within two organelles, chloroplast, mitochondria, and on the rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome. This helps in the cell division. Centrosome is like this, a cylindrical organelle which is helping in the cell division. Cells can vary greatly in size, shape, activity. Here I have shown few example of cells. See, this is the red blood cells. This is the nerve cell, and these are actually the longest cell. These are the white blood cells. These are the columnar epithelial cells. Now here comes trachid and mesophyll cells. This trachids, mesophyll cells, these all things again I have discussed in my earlier chapters, okay? In plant morphology and plant anatomy chapter. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary in shape like disc-like, polygonal, columnar. Here we can see the several shapes, cuboid, thread-like. And the shape of cell may vary with their function also. Now we will come to the final section of our today's video, that is prokaryotic cells. Prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and pleuropneumonia-like organisms, or we call them PPLOs. See here, there is bacterial cells, here there are PPLOs, and there are viruses. Now, they are generally smaller and multiply more rapidly than the eukaryotic cells. What? The prokaryotic cells are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. 4 basic shapes of bacteria, this also have been discussed in the previous chapter. Bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spirillum or spiral. All prokaryotic cells have a cell wall surrounding the cell membrane except in mycoplasma. Here also we know there is a fluid-filled matrix called cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, that is the single chromosome or circular DNA, many bacteria have small circular DNA, which is known as plasmids. Later on, when we will study biotechnology and molecular biology, we will find that there is a huge use of this plasmids. Okay, so it is written that smaller DNA are called plasmids. Plasmid DNA confers certain unique phenotypic characters to such bacteria. So, what is such character? That is resistance to antibiotics. Plasmid often offers resistance to the antibiotics. Nuclear membrane is found in eukaryotes but not in the prokaryotes. Likewise, no organelles are found in prokaryotic cells. Prokaryotes have one unique thing, that is inclusions, or we call it as inclusion bodies. Prokaryotes have a specialized differentiated form of cell membrane called mesosome, which is a characteristic feature of the prokaryotes. They are the infoldings of the cell membrane. Now let us come to cell envelope and its modifications. Cell envelope consists of tightly bound three-layered structure, that is glycocalyx, cell wall and plasma membrane. They act together as a protective unit of the cell. Bacteria can be divided into two groups or classified into two groups on the basis of differences in their cell envelopes and the manner in which they respond to the staining procedure, which is the gram stain. The envelope that takes up the gram stain are called gram positive, and which do not are known as gram negative. Glycocalyx differs in composition and thickness among different bacteria. Loose sheath called slime layer in some, and in some it is thick and tough called capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacteria from bursting or collapsing. The plasma membrane is, the plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. Prokaryotes, here we can see, form a special membranous structure called mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They are basically responsible to increase the surface area of the plasma membrane and the enzymatic content. In some prokaryotes, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. The one which are motile will contain filamentous extension called flagella. Now, what is flagella? Flagella is composed of three parts: filament, which is the longest portion, then hook, and finally the basal body. It looks almost like this. This is the basal body, from which this is the hook, and this is the filament or the longest portion. Clear? Now there are other structures called pili and fimbriae, which are basically their anchoring organs. Okay, so they help the bacteria to attach to rocks in the streams and also to the host tissues. This is done mostly by the fimbriae, but they do not play a role in motility. Okay, pili are elongated tubular structure made of special proteins. Now let us come to ribosome and inclusion bodies. Ribosomes are associated with plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and made up of two subunits 50S and 30S, which when present together forms 70S, that is the whole prokaryotic ribosome. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into the proteins. So, here we learn about the ribosomes. Now let us come to inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. They are not bound, we must remember this, they are not bound by any membrane system and lie free in the cytoplasm. For example, phosphate granules, cyanophysin granules, or glycogen granules. Gas vacuoles are found in the blue-green and purple-green photosynthetic bacteria. So here we end our today's session, that is cell structure and function till the prokaryotic cells. In our next session, we'll continue with the eukaryotic cells. So if you like the video, please press the like button, share and subscribe the video. Till then, happy learning. Thank you. Bye-bye.